Hello, and welcome to this webinar titled Regulatory Strategies and Case Studies for Rapid Sterility Testing of Gene and Cell Therapy Products, hosted by Biopharma Asia magazine and presented by Dr. Michael J. Miller, President of Microbiology Consultants, LLC, and Laurie Dane, Pharma Microbiology Scientific Director at bioMérieux. My name is Stephen Edwards, and I'll be your moderator. Now please allow me to introduce our first presenter. Dr. Michael J. Miller is an internationally recognized microbiologist and subject matter expert in pharmaceutical microbiology, contamination control, and the validation and implementation of rapid microbiological methods. He is currently the president of Microbiology Consultants, LLC, and the owner of rapidmicromethods.com, an educational website dedicated to the advancement of rapid microbiological methods within a variety of healthcare, pharmaceutical, consumer, and related industry sectors. For 30 years, he has held numerous R&D, manufacturing, quality, business development, and executive leadership roles at Johnson & Johnson, Eli Lilly and Company, and Bausch & Lomb. In his current role, Dr. Miller consults with multinational companies in providing technical, quality, regulatory, and training solutions in support of rapid methods, sterile and non-sterile pharmaceutical manufacturing, contamination control, remediation, environmental monitoring, sterilization, and laboratory operations. I'll now be handing over to our first presenter. Welcome, Dr. Miller.
Thank you. I would like to thank everyone who is currently on the line. I do appreciate your time. And today I would like to discuss the regulatory strategies and some case studies on rapid sterility testing for gene and cell therapy products. These are ATMPs. And what I would like from the moderator, I'm just having some problems advancing the slides, so if you would be so kind to be able to do that for me, I think we'll be able to make the webinar work just fine. So if we could advance to the first slide, please. ATMPs, advanced medicinal products for therapy, cell therapy, gene therapy products, are engineered cells or tissues administered to patients for treating a human disease. These can also be gene therapy in terms of transfer of materials such as DNA or RNA into the cells of the patient's body, as well as somatic cell therapy, which is the transfer of intact live cells into a patient. And this is one of the areas which is growing fairly quickly, both in the U.S., in Europe, and in Asia. If we can go to the next slide, please. I want to focus for a moment on somatic cell therapy. The cells are going to be originating from either the patient, which is an autologous cell preparation, or can come from human donors or animal donors. And what happens is the cells are going to be modified in a laboratory in a controlled environment before those products are administered back to the patients. And one of the areas where we have the most growth in this area is chimeric antigen receptor, or CAR T cell therapy. If we can go to the next slide, please. Thank you. Now, CAR T therapy is a process by which a patient's own immune cells, and this starts with the patient's blood, is going to be collected, and then we can purify, we can capture the T cells and modify those T cells such that we are going to express a receptor on those T cells, the surfaces, and those receptors are going to be very specific for recognizing cancer cells or tumor antigens within the patient's body when those transformed cells are being infused back into the patient. And once those cells go back into the patient, if the receptor binds to the tumor antigen, the T cell is going to be specifically stimulated to attack and kill those cancer cells. Okay? Now, in 2017, 2 companies received FDA approval for CAR T therapies. They include Chimera and Yescarta, and Kite Pharma approvals for both B-cell acute leukemia and large B-cell lymphoma. We can go to the next slide, please. On this slide, what you are seeing is an overview of the manufacturing process for CAR T-cell therapies. Now, this is one example. There are different ways of creating these transformed cells, but generally speaking, an apheresis sample, the blood sample taken from the patient, is going to be brought into the manufacturing area or the laboratory where we will isolate the T cells. We will then transform those T cells by inserting the gene specifically to transform the T cell to recognize the cancer cell once it's reinfused into the patient. Once we do this, then we're going to grow millions of copies of those T cells, and then once that is done, we will reinfuse those T cells into the patient, and then the T cells will do their job. Next slide, please. This is an example, a scanning electron micrograph of a CAR T cell that is attacking a breast cancer cell. So it's very specific, and the results are rather rapid. Next slide, please. But there are challenges in working with gene and cell therapy products, especially CAR T-cell therapies. For example, most of these types of products have a very short shelf life. Okay? This is unlike conventional pharmaceuticals, even large molecule pharmaceuticals, where the shelf life can be for months or years. These are usually products that need to be reinfused into the patient as early as, within days. Also, the administration usually occurs long before the results of a conventional sterility test can be achieved, which in most cases is at least 14 days. Many of these types of products would also render the sterility test medium turbid, and for that reason, the sterility test would actually go longer than 14 days because you would need to subculture. Another challenge with these types of products is we have low production volumes. Okay? We're not talking about hundreds of thousands of vials. We're not talking about 30,000 liters of product in a compounding tank. These are very, very low volumes, and for most cases, we cannot meet the compendial requirements for sample size, for the number of vials and the amount of product from each vial. And what happens is, if we use more product for testing, then we have less product that goes into the patient, and that lowers our success rate for a positive clinical outcome. Now, the question becomes, what can we do in terms of sterility testing these types of products? And if we make alterations to our current strategies, for example, the compendial testing strategies, what do the regulators say about this? And if we can go to the next slide, I'm happy to share with you some very recent policy changes from the regulators that allow us to develop alternative strategies for these purposes. In 2012, the FDA published within the Code of Federal Regulations new sterility testing strategies and policies for biologics. Okay? They amended the sterility testing requirements in its final rule, and I want to point out some of the key changes from an FDA perspective. First, the FDA had stated that there are new alternative and new rapid methods, including ATP bioluminescence, solid phase cytometry or chemiluminescence, and even the measurement of carbon dioxide in sealed containers as new methods by which we can obtain sterility testing results much faster than the compendial 14-day sterility test, and that we as manufacturers can benefit from using these types of methods. Next slide, please. within that same policy change, the FDA stated that any new sterility test would have to be validated to make sure that we are, in fact, detecting the presence of low levels of viable microorganisms, and that any new method that is non-culture based, one that would not normally utilize conventional media, or those that are going to change significantly from what's listed in the compendial test, such as USP 71, then we require validation, for example, for limit of detection, or LOD, for ruggedness, robustness, and the ability to detect a wide variety of microorganisms, or specificity. Next slide, please. More recently, in the summer of 2018, the FDA came out with its draft guidance specifically on gene therapy. And in this guidance document, the FDA lists some alternative and rapid methods which may be needed for testing these new types of products, which include rapid sterility testing, rapid tests for the detection of Mycoplasma, including PCR-based tests, as well as rapid endotoxin tests. Next slide, please. Within this draft guidance, the FDA also recommends that when you want to validate these new methods, you need to ensure that you demonstrate an equal or greater assurance that the new method, when compared to the compendial method, is going to be reached. And you need to do this before you license the actual product. So I'm going to talk a little later on about some case studies working with some clients within this space. But most of us understand this to mean that during clinical studies, you don't necessarily have to complete your entire validation program. At the very least, you need to demonstrate method suitability and some amount of testing to demonstrate you will detect the positives. But prior to licensure, prior to the final approval and the use of the product routinely with patients, you need to complete your full validation program. Okay? The FDA in this guidance document also describes alternative methods in the event that you need to have a result prior to a sterility result on, for example, the final product. For example, you could do an in-process sterility test taken on samples about 48 to 72 hours prior to final release. Another recommendation is that you can use some other rapid detection test or a sterility test or even a gram stain prior to the results from a finished product sterility test to give you an idea if you have some growth or gross contamination. Okay? Can we go to the next slide, please? Now, these are some of the recent changes from FDA. If we look at the European Union, there has been a recent guideline published for ATMPs, and this is listed on the slide that you have in front of you now. This new guideline became effective in May of last year, and it applies to the manufacture of ATMPs that are either used in a clinical trial or those that have already been granted a marketing authorization for sale in Europe. Next slide, please. I'd like to point out some of the more relevant sections within this EU guideline for ATMPs. One of them states that if you apply the sterility test for finished product, okay, for example, what you would find in the European Pharmacopoeia Chapter 2.6.1, that chapter and following the chapter in terms of testing timeline to result, as well as the amount of product required, may not always be possible because the EU guideline understands and states that material may not be available to meet those compendial requirements, or you may need to reinfuse that product prior to the results of a finished product sterility test. And in those cases, it is appropriate to adapt an alternative sterility testing or sterility assurance strategy. Okay? This may include using alternative methods, either on the finished product or using preliminary results, such as upstream samples of testing spent media, or even intermediates or other in-process samples. Okay? So this is something similar to what the FDA guidelines had already published. Next slide, please. The EU guidelines also state that if you're testing intermediates or in-process controls instead of or in addition to the finished product, you need to make certain that whatever the results are for those in-process samples are going to be relevant to the type of results you would normally expect if you did a sterility test on the finished product, and that makes sense. The guideline also states that you can use validated alternative or rapid micromethods according to another chapter within the European Pharmacopoeia, Chapter 2627, as long as you perform method suitability on the product. So if we go to the next slide, I can briefly discuss what is stated in Chapter 2627, which became effective in the summer of 2017, the microbiological examination of cell-based preparations. Here we find a similar theme as what I've been discussing so far, that shelf life of cell-based preparations, including ATMPs, may not always allow you to perform a conventional sterility test prior to administering that product to the patient. Okay? And that there may be technical or sampling issues, as we discussed earlier. Many of these products, if it's added to a conventional liquid medium, would render that medium turbid, and we would have to subculture, and then we'd end up with more than a 14-day sterility test. Okay? And certainly the amounts available for testing. Next slide, please. So this chapter in the European Pharmacopoeia understood that there was a sampling restriction for these types of products and came up with the following recommended alternative sampling plan. For example, if the total product volume in a batch of ATMP was between 10 milliliters and 1,000 milliliters, the minimum test sample volume would be 1% of the total product volume. And that 1% of the total batch would be divided between, if you used a conventional sterility test, the aerobic and the anaerobic bottles. Okay? If the total batch size was between 1 and 10 milliliters, then you would only test 100 microliters, that being divided into the aerobic and the anaerobic bottles. If the batch size, if the total batch for this type of an ATMP was less than 1 milliliter, then obviously there would not be sufficient volume to do a test, and you would rely on testing upstream or intermediate samples or spent media to give you an understanding of what the potential would be for a contaminated finished product, even if it was less than 1 mil. Okay? Next slide, please. More recently, the USP came out with a proposed informational chapter, numbered 1071, called the Rapid Sterility Testing of Short-Life Products. Okay? This is going to be finalized later this year. And this is a chapter that is intended to provide the end user with guidance when a compendial sterility test is just not suitable for product release. And they've identified a number of stakeholders, not only ATMP or gene and cell therapy manufacturers, but also compounded sterile preparation, compounding pharmacies, as well as PET products. Okay? Let's go to the next slide, please. And we'll take a closer look at what's been proposed in Chapter 1071 with the USP. The USP states, and this is going to be very similar to what the other guidance documents have already published and what we've discussed today, that the current 14-day sterility tests are not suitable for short-life products, especially those that are intended for immediate administration, that a rapid sterility test should be considered and should be risk-based, and that those stakeholders, the ones that I had mentioned in the previous slide, should decide on the best technology based on their user requirements, which could be the time to result, the specificity or the types of organisms that are required to be detected, the limit of detection, the sample size, and possibly other product attributes. Next slide, please. Interestingly, the chapter refers to the European Pharmacopoeia Chapter 2.6.27 and the 1% sampling plan as well. Now, this is an informational chapter where we're not going to see any full harmonization between this chapter and what's in the European Pharmacopoeia, but it's good to know that the various pharmacopoeias are thinking similarly in terms of sampling plans for these types of products. The chapter also states that other rapid microbiological methods may be used as an in-process control prior to releasing the final product using a finished product sterility test if we want to understand whether there is gross contamination upstream that may be relevant to what we might find if we performed a finished product sterility test. So the USP is not only talking about a finished product sterility test, but is also discussing the potential use for these new types of methods for doing in-process or upstream sampling. Next slide, please. I'd like to close out my review by sharing with you some case studies. These are based on my work with a number of cell therapy, gene therapy companies over the last few years in working on validating rapid sterility testing using some of the commercially available systems that are now available. And what we first did was looked at what the user requirements were. For example, what sensitivity do we want to be at? Do we want to be at a single cell detection capability or something else? What's the required time to result? Can we go 7 days? Do we need to go 4 days, 3 days, and try to, or even sooner, and try to match an appropriate technology? And also to determine whether the products that we were working with were going to be compatible with the various rapid methods. Some rapid methods for sterility could be a direct inoculation, or they may require something like a membrane filtration. And sometimes these cell cultures are very difficult to pass through a membrane, and we need to develop alternative preparations for those cell products in order to pass them through a filter without impacting the ability to detect the viability of contaminating microorganisms. So all of these types of attributes, the product attributes, the technology attributes, need to be taken into consideration when deciding on the best possible rapid or alternative technology for a sterility test. Okay? Also, we pass this information to the relevant regulatory agencies. For example, a Type C meeting with the FDA to get their buy-in on the validation strategies, the use of a particular technology as well. And then you perform some feasibility work with the supplier, you validate the method, you do method suitability or product suitability studies, and then finally demonstrate equivalence with the actual compendial test to demonstrate that the alternative method, what we want to use, is in fact no worse or non-inferior to what we would normally find if we were able to use the compendial assay. Next slide, please. So what we did for a number of my clients is we considered doing these upstream or in-process samples. In some cases, no limitation to the available sample size, for example, spent media or rinse fluids. The only challenge there is we had to demonstrate that if we detected microorganisms in those upstream samples, those results would be relevant to what we would normally find in a finished product sterility test. Sometimes it was a little difficult. In other times, it was relatively easy to understand that, for example, with spent media, in one of the upstream steps, If that's contaminated, there's a high likelihood that that contamination would be passed down to the finished product. Okay? Regardless of whether there was some upstream sampling or not, most of the companies I've worked with chose to test the finished product. Okay? We also considered testing residual product from the bulk vessel prior to filling, because there will always be some amount of material left in that vessel, even if it's a small vessel, and you can do this assuming there is no risk or a low risk of contaminating that material in a downstream process. So, for example, you can have a filling vessel, but if you have any type of an open filling process, even in a controlled environment, there is always a risk of contamination. You may be able to do a risk assessment and justify that that risk is negligible if all of your connections are going to be closed. Okay, if you're using some welds in your connections and making those welds in an ISO 5 or a grade A environment, then you may be able to justify doing that upstream work and not relying so much on the product that has actually been filled into vials or cartridges. That, again, is up to you to do that risk assessment. Next slide, please. This is a little busy slide, but it just gives you an understanding of the different manufacturing points and the different phases in a CAR T cell manufacturing process. If you focus on the green arrows, these are some areas where you might be able to do some testing upstream that is relevant to the overall process. For example, when you expand the T cell in media, or there may be some spent media or wash fluids. Many times we're going to concentrate the cells, and there may be some leftover supernatant from those concentration steps that you might be able to test, assuming that those results would be relevant to the cells that are going to be further processed. And then, of course, you can do the final product testing. Sometimes the final product in these types of configurations are going to be cryopreserved, which may give you a little longer shelf life. You can do the testing on cryopreserved product, or if it's needed immediately, on the fresh product if it's not being cryopreserved. Next slide, please. So as far as sample size is concerned, this is where I'd like to close out my presentation and give you some examples of where the challenges are and how you can follow the compendial recommendations for sample size. So one of the first questions you should be asking yourself in this space is, if we are making a cell therapy product and we do have low amounts of finished product available, can we meet the compendial sterility test requirements? So I want to give you two examples. The first one is on that top row, where the product is filled into 4 IV bags. Each IV bag is a 60 mil fill, so the total batch size is 240 milliliters. If we follow the compendial harmonized sterility test, the sampling requirements state that we must test 4 containers. Okay? Now, we already have 4 IV bags, so the compendial test would want us to go into each of the 4 containers, and the minimum quantity for each medium would be 20 milliliters from each bag. So 20 milliliters times 4 bags, okay, times 2 different media, that's a total of 160 milliliters, okay? That's pretty much impossible to do, because we would lose 67% of the batch. We have to go into every bag. That would ultimately need to go to the patient, potentially increasing the risk of contamination. Okay? We look at the next example. I want you to assume 15 vials in the batch, 4 mil fill. The total of the batch is 60 milliliters. We would still be required, according to the compendial sterility test, to sample each of the 4 vials. The minimum quantity for each medium would be 2 milliliters from the 4 vials. That would be 16 mils total. Okay, the loss is not as great. It's 27% of the batch, but in order to reduce the potential for contamination, we would probably sacrifice 4 of those vials. Now, it's up to the end user to decide in this point, can we lose 27% of the batch and still have enough to infuse in the patient and not have an issue in terms of a clinical outcome? I can't answer that for you. This is something for you to do. So here are some of the real-life challenges that I've worked with some of my clients. And if we go to the next slide, I'll give you an understanding of how we follow the compendial recommendations in European Pharmacopoeia 2627 and now the proposed Chapter 1071 from the USP. We're going to use those same scenarios with the IV bags and the vials. So let's take a look first at the 4 IV bag scenario. The minimum sample size, if we assume 1% of the batch, is going to take 2.4 milliliters from a single bag. Okay? Now, the potential risk here is we're going to sample into a single bag. Okay? We don't want to lose the rest of that material. That's close to, what, 58, 57 milliliters from the bag. So we need to be very careful about taking that 2.4 from the one bag. But if we look at the batch loss, that's only 1%. Okay? Now, we could test a greater volume if one bag is going to be sacrificed. If we sacrificed one bag, that's 25% of the batch, we'd have 3 bags left. Okay? That's going to be determined whether or not there would be a risk to the patient or not. Okay? And that's something that's going to be very specific to your process, how much material you have in the finished batch and how many containers or bags that you might have. But it's something for you to consider. The second scenario on the bottom, 15 vials with a 4 ml fill. We have 60 ml batch. We would take 0.6 milliliters from one vial. So we're sacrificing one vial. The batch loss is 7%. It was determined that that is an acceptable loss where we wouldn't lose any clinical outcome in terms of the amount of product that we need to be re-administering into the patient. We could test, therefore, a greater volume from that one vial. Remember, it's a 4 ml fill, so instead of just doing 1% and testing 0.6 mils, we can double it or triple it. We can test a bit more and even have more material left over from that one vial to do other types of tests, okay? Mycoplasma testing or have some retain. So there are some options here. At the end of the day, you need to look at what your product is, how many containers, what's the fill volume, what you could afford to lose in terms of product performance and patient safety, and go from there. We go to the next slide, please. So obviously, I've summarized so much information, and I want to point you to an online resource where you can review at your leisure much more information regarding the testing of cell therapy and gene therapy products, ATMPs, sterility testing, for example, on a website called rapidmicromethods.com. Here you will find the most up-to-date information on the regulatory and compendial policies, especially the ones we've discussed today, and guidance on how to validate these alternative methods and whether they may present you with a good return on investment. The website also contains an extensive list of published references, as well as all of the regulatory guidelines that I've discussed in my presentation today, so you can click on those links and go directly to those PDFs or the regulatory websites. Also, to help with selecting the most appropriate method that you may want to use for rapid sterility or even upstream sterility or bioburden assays, there is a product matrix that walks you through workflow, limit of sensitivity, detection, quantitation as well. And there are also a lot more information on industry news and a blog on the newest technologies that are being developed that may also be relevant to what we've discussed today. Okay? We go to the next slide, please. So I thank you for your time. I know we will have some time later in this webinar for questions and answers, but even after the webinar is over, I encourage you to contact me if you have some additional questions from what I've discussed today. My contact information is on the slide. One of the easiest ways to get to me is send me a text or a phone call. My number is very easy to remember. It is +1 rapid. So again, I appreciate your time today, and I will now turn the program back to our moderator.
Thank you, Dr. Miller. Once again, I would like to remind our viewers that there will be a live question and answer session at the end of the webinar. Please feel free to send your questions at any time during the webinar via the questions tab located directly below your webinar screen, and we'll go through these at the end of the presentation. Now please allow me to introduce our second presenter. Laurie Dane is the Director of Scientific Affairs at bioMérieux and has experience in clinical, environmental, and industrial microbiology. She is a technical expert on rapid and alternative methods and participates in the sourcing and evaluation of new technologies and potential partnerships in the field of microbial control. She provides scientific support to the healthcare business in North America and is responsible for managing feasibility testing and method development for bioMérieux instruments and culture media products. Laurie received her PhD from the University of Minnesota in microbial ecology. She also holds a master's degree in limnology from the University of Minnesota and a bachelor's degree in medical technology from Ferris State University. I'll now be handing over to our second presenter. Welcome, Dr. Daane.
Thank you. Hello, everyone. First, I would like to thank Dr. Miller for his informative presentation. I think he provided a really nice detailed update on regulatory guidelines for cell and gene therapy products. So in my presentation, I'm going to focus on the why of a rapid method, specifically why implement a rapid method for cell and gene therapy products. And the most important reason is to improve patient care. Now, Dr. Miller also defined advanced therapy medicinal products, also called ATMP, and detailed cell therapy, specifically outlining CAR T-cell production. But before we begin, I really want to take a moment to stress how cell and gene therapy, it isn't just another pharmaceutical product. I don't think we can look at these products the same way we look at traditional pharma or even bioproduction products. These products are life-altering. They are many times a person's last chance of survival, and there are many case studies that can only be described as miraculous. There may be people on this webinar that have been personally impacted by cell and gene therapy products themselves. And the people working in this industry are fully aware that what they're doing that they're saving lives. And when you go to conferences and you have discussions with them, you can really hear it and you can feel their passion. So I just wanted to say that this is really such an evolving market, and it has an incredible impact to people's lives. So this slide is really showing a very highly simplified needle-to-needle supply chain cycle. And we know that there are numerous challenges associated with manufacturing cell and gene therapy products, which I will summarize shortly. But for these products, the supply chain activities must be highly synchronized and managed to successfully deliver the right product to the right patient in the right condition. Again, all of the testing and the manufacturing for these products are really to ensure patient safety and to deliver a product to this patient in the best way possible. Dr. Miller also touched a little bit on the differences between allergenic and autologous, but from a manufacturing point of view, the differences really highlight the need for enhanced chain of custody and chain of identity. So for allergenic products, one batch serves many patients, and the product may also be cryogenically stored for weeks to months, and sometimes This is called scale up. So, however, for autologous products, one batch serves one patient. So this seems really easy if you were a manufacturing facility and you just had this one product, or maybe even if you had 10 products all at once. But what if this one individual really were to equal 1,000 products? How would you manage 3,000 products or 7,000 products at a time? So therefore, the biggest challenge when I talk to people in cell and gene therapy is chain of custody, data integrity, and the need for electronic tracking. And the FDA has stated many times that they really focus and look at where there are the risks of cross-contamination. So when you're performing your testing, closed systems and using test methods that can provide the best data integrity as possible is key for managing the supply chain of autologous cell therapy products. So just to summarize the challenges of cell and gene therapy, clearly it's usually a very low production volume, very short shelf life. It could be just maybe only a couple weeks, or it might even be days. They're very sensitive to temperature, and you have to maintain sometimes a cold supply chain. There could be complex raw materials. The starting material itself can be highly variable. The analytical methods used to test the samples are also very complex. You have to have enhanced tracking and data integrity, and you have to have the ability to scale up. So scalability, it's all based on patient needs, and so that's a huge challenge for cell and gene therapy companies. But again, the importance of all of the handling of this and handling these challenges is to get the right product to the right patient in the right condition. This is very, very critical. So coming back to our cell therapy production process, the challenge to quality testing, also called the QMS, is that it's a continuous process. You can't, there's no in process really. It's just continuous, and it takes days. It's not months anymore. This process might be just days. And a few examples that I heard at a recent conference showed a typical production cycle of 14 to 20 days. And because the goal is to get the cells back into the patient as soon as possible. So the quality management system includes testing for bioburden, viral clearance, water, environmental, mycoplasma, sterility, and endotoxin. Now, these products cannot be terminally sterilized. So they're usually processed following aseptic techniques. So there's no time for end product testing following the traditional methods. So cell and gene therapy companies are following the guidelines that Dr. Miller outlined, and they're taking a risk-based approach. Many companies are actually even using a PAT approach, which is an approach that is generally used for terminally sterilized traditional pharma. So what they're doing is the manufacturers are doing all of their testing on raw materials, and they're implementing a very robust environmental monitoring program to manage their facility. So an additional challenge is that the majority of testing is performed in a separate centralized QC laboratory. There is a lot of value in finding opportunities for rapid testing and also bringing this testing out of the lab and directly to the production floor. And the best way for this to happen is to work with suppliers that offer rapid solutions. So in my presentation, I'm going to focus on two rapid testing solutions that are ideal for cell and gene therapy. The important thing is that they are simple, they're simple to use, they're fast and accurate. Now, the previous presentation from Dr. Miller provided detailed information on all of the available regulatory guidelines, and he also provided a nice link for you to be able to get your hands on all of the guidelines yourself. But here I'm described the three main types of rapid microbial methods that can decrease the time to result from the 14-day compendial test that's described in both the EP and the USP. So alternative methods, they have been embraced and they've been described in 516 in the European Pharmacopoeia and in the USP 1223. And these again include growth-based methods like CO2 production and bioluminescence, direct measurements such as solid phase cytometry and flow cytometry, and lastly, cellular component analysis. More recently, the EP has published 2627 to address microbiological examination of cell-based preparation that includes rapid methods. And there's also the new chapter. Again, that the exciting thing is that it will be published, I think, next month and will become official at the end of this year. And this is specifically for rapid testing of short shelf life products. Now, what I've seen is that I think there's been a big shift from rapid methods being seen as an alternative method to the compendial test, but to now the compendial test being a rapid method. So rapid methods can reduce the time to detection from 14 days to as little as 4 hours. So that's been an incredible evolution, and I think that kind of goes hand in hand with the needs of the industry because of the evolution of what's happening in medicine today. So the first rapid method that I'm going to discuss is one that is a growth-based rapid method technology based on CO2 production that is ideal for cell and gene therapy products, and it's the Baxter 3D Dual T system. The Dual T has both low and high temperature incubation modules, allowing it to closely follow the Pharmacopoeia guidelines. There are also other models that are available for single temperature incubation. And what's nice about the system is that it's modular in that you are able to add additional drawers that are controlled by a single control unit. Now the product sample is added to aerobic and anaerobic bottles and then placed directly into the instrument. So once the sample is added to the bottle, you have full data integrity. And I've talked to a lot of cell therapy companies that this is what they love about the system, is that it is so easy that they're able to put the product directly into the bottle, put the bottle into the instrument, and the instrument does everything else. And they don't have to worry about removing the enriched sample or doing additional testing on the product. So the BTA system utilizes a colorimetric sensor and reflected light to monitor the presence and production of CO2 that's dissolved in the culture media. So once the bottles are loaded, the colorimetric sensors, they're scanned every 10 minutes. So when microorganisms produce CO2, as they metabolize the substrates in the culture media, and this changes the color of the gas permeable sensor that's in the bottom of the culture bottles from a blue-green color to a yellow color. So once growth is detected, the system alarms both audibly and visually, and the sample data is recorded. Now, the bottles that are shown here with the sensors, with the yellow on the left and the blue-green or gray on the right, that's a very extreme sample. I can tell you from experience that I have seen positive results from bottles that contain that were spiked with Clostridium sporogenes, for example, where the bottles became positive in less than a day. And with at least the human eye, I was not really able to discern the difference between the sensor colors. That's how sensitive the BTA system is. So the big advantage of the Baxter system is that it's a medical method. So the bottles are continuously monitored with the bottle sensor being read every 10 minutes. And this continuous monitoring allows for faster detection of positive results. And the reading is objective and completely independent of turbidity, so there's no need for performing subculture. Now, since the BactAlert has been introduced many, many, many years ago, there's been numerous publications using the system for both traditional and cell-based products. But recently, the NIH published in the Journal of Clinical Micro in February of this year, comparing the traditional and automated culture systems on 118 different microorganisms. I think this is probably the most comprehensive study that I've seen, and the results showed that the BactAlert system can be used as an alternative sterility test. Now, with that said, most cell and gene therapy companies are not testing 118 different isolates and focusing on environmental or slow-growing organisms. They're more or less focusing on the clinically significant organisms and organisms that are able to be detected usually within 2 to 3 days. Now, the benefits of implementing a rapid sterility test, such as the BactAlert, include the use of automation that improves work efficiency and data integrity, but it also provides standardization, ease of use, and objectivity. It also allows the testing to be performed closer to the production floor. There's no need to have an isolator, for example. Now I would like to shift to the second rapid testing, and that is Mycoplasma. So just to give everyone an overview of the challenges of this microorganism, it's ubiquitous in the environment, and it can be found in raw materials as well as being brought in on personnel. It's considered a parasite and also can be a commensal. They don't always kill their host cells, and they can go easily unnoticed. And the media in which the product itself is generally made is a perfect environment for these for Mycoplasma to grow, and the quality and quantity of the product is decreased, and some Mycoplasma are clinically significant. So Mycoplasma is very difficult to prevent, it's difficult to detect, and it's very, very difficult to eliminate. So in cell and gene therapy, so what's nice is you really, for this particular market, you really need a fast, simple, and robust solution. Because again, you're dealing with a product that has a shelf life of much less than 14 days. And when you look at the traditional method, it really can't be performed in this particular market because it takes 28 days in order to get a result. And it's very arduous and requires a lot of skill. The second method, which is a traditional method, is the indicator cell method. Now, while this method is slightly faster, the results are in 5 to 8 days, and it still takes a lot of technical experience. There are faster methods out there and that are based on molecular systems are available, and they can provide results in about 5 hours. But the test method still requires quite a bit of expertise. Many times a molecular biologist has to be on staff. It requires a specialized laboratory, and it can be prone to contamination due to the open test system. So while I discussed previously with the BactAlert system that you can cut the time in half compared to the traditional test method, what if I could tell you that if you could take a 28-day test and turn it into an hour and make it so simple that anyone can perform it. Plus, it doesn't require a special lab, and it could be performed potentially directly on the production floor. I would like to introduce the lab in a pouch. It's a completely closed system for performing molecular mycoplasma that will take less than 2 minutes of hands-on time and provide results in about an hour. It is so simple it can be performed by anyone, anywhere, and any time. This is an overview of the different steps that are involved. Again, the product sample is just pipetted into a vial that then loads the pouch. The pouch then is placed into the system. The system does the testing, and a report is generated, and it's a presence-absence test for Mycoplasma. So the benefits of rapid Mycoplasma testing are the same as for rapid sterility testing. The main objective is speed and decreasing time to result, cutting sterility time to detection in half and bringing Mycoplasma testing from 28 days to 1 hour. So both solutions can be so easy they can be brought out of the lab and close to the manufacturing where they are needed. So just to summarize, cell and gene therapy production is highly complex with numerous challenges. There's a need for enhanced chain of custody and data integrity to ensure the right product is delivered to the right patient without cross-contamination. There are new regulations which allow risk-based approach for microbial sterility and Mycoplasma testing, and new alternative methods are moving the lab to the production floor. With that, I thank you, and I would like to give a special acknowledgement to my colleague Brian Whitehouse for helping me put together the slide deck and providing my contact information. If any of you have any questions, please feel free to contact me. And with that, I will hand over to our moderator.
Thank you, Dr. Daane. Now, we will begin the question and answer segment of the webinar, but once again, I would like to remind the audience that you can still send your questions in via the questions tab located directly below your webinar screen. So now our first question is, with the 1% sampling, does it have to be the same cell concentration as the actual product, or could we dilute the sample with the freezing mixture that was used to formulate the final product to increase this volume? Dr. Miller, would you like to answer this?
Yes. So this is a good question. The 1% sampling plan is related to the total batch size of the finished product in its final containers. Okay? Which means if you have a cell concentration count in the finished product, it is assumed that the 1% sampling is going to be relevant to whatever that final cell concentration is. Now, that being said, to look at the second part of your question, if you are able to take 1% of the total batch size, you can dilute that down, whether you're using the cryopreserving fluid or some other type of a diluent. You can dilute that down to make it easier for the testing. For example, if you want to pass this through a filter or do some other sample preparation work. So you can further dilute that, whatever the 1% is, even if it's going to impact the cell concentration. But at the end of the day, the most important volume to use for your finished product testing would be 1% of the total volume of the final batch size.
Thank you. And Dr. Daane, do you have anything to add with that?
I'm sorry, I was on mute. No, I think that Dr. Miller answered it very well.
Perfect. Our next question is, what is the best rapid technology to use for sterility testing of cell therapy products? Dr. Miller, would you like to take this again?
Yes, well, it depends. There's no right or wrong answer here. It's the selection of a particular method or a technology is going to be dependent upon your user requirements. For example, what is the level of sensitivity? Okay, because there are systems that can get down to single cell detection either after an enrichment phase or a direct measurement, like a solid phase cytometry. So that's one example. Number two, what really is the time to result? If you can afford a 7-day test, then there are growth-based methods that can be validated to give you a comparable result to the compendial assay in 7 days, or 3 days, or 2 days. Or if you need single shift, the same day results, there are non-growth-based systems that may work. Okay, so that's something else. Number 3 is whether your sample is going to be compatible with the system you'd like to use. There might be some incompatibility issues. I mentioned one earlier in my presentation, is that there are methods you could use to break apart a human T cell or even a B cell to where it passes through a filter and not impact the viability of a microorganism that may be present. But that may not be always the case depending on what your total formulation is. So my recommendation there is, before you make a final decision on a technology to use, is to work with the supplier of the method, send them some of your samples so they can do some false positive and false negative testing and see if your matrix will actually work in that system. If all the planets aligned and there's no incompatibilities and the system can meet your user requirements or most of them, then that's the point where you say, I have a good probability of success of using that particular system. So that was a more detailed answer to a quick question, but we all need to understand that there are a lot of things that go into making a final decision on what would give you the best probability of success going forward.
Thank you.
I would like to add
can
I add a little bit on that one? Yeah, I agree that in order to find the right rapid method, you really have to look at your product matrix and compatibility. Many times, especially in cell and gene therapy, the product's not filterable. So you end up having a situation where you need to do a direct inoculation or a direct test. And what's great about the BacT-Alert system is that it does allow for a direct inoculation of the bottles. Compatibility is very good with cell-based products. That's the other thing, is a lot of times different systems, when you have a lot of cells that are present, it could interfere. The BacT-Alert system has been used for testing cell-based products for a number of years, well before cell and gene therapy. And for sensitivity, the LOD is generally if a cell was present and inoculated in the bottle, it will grow to a point in which the system will detect a positive. Just a couple weeks ago, I did have a conversation with someone who is using a BacT-Alert system, and I asked directly, how long are you able, or do you incubate your products? And they said, well, we keep the bottles on the system for 5 days, but we generally release the product for infusion at 4 days, or even before the bottle results are off. And they've been validating to clinically significant organisms with positive detection in less than 3 days with all of the organisms. So they're moving more towards a 3-day growth-based test.
Just to, and let me just come back and add, I had mentioned in my response earlier that there certainly are challenges with working with many cell therapy matrices. In some cases, they can't be filterable, but I also have some clients that I'm currently working with where there are ways of destabilizing human cells or mammalian cells to pass through filters, which do open up areas for a wide variety of systems, including solid phase cytometry and other systems that utilize filtration. But it all comes down to working with the supplier during feasibility studies to determine whether or not these types of systems are appropriate. And if for some reason you can't get things to filter, you may not have much of a choice but to go to something like a direct inoculation platform.
Thank you both for your comprehensive answers. Our next question is, has the lab in the pouch been tested for sensitivity of Mycoplasma at 10 CFU milliliters or lower? And how many species of Mycoplasma has it tested? Will there be matrix interference with culture media? So obviously, Dr. Daane, would you like to answer this?
Well, those are a lot of really good questions. I mean, it is a new product that will be released later this year, which we're very excited about. We've done quite a bit of testing and validation, and we're currently doing equivalency with by a third party for comparing it to the compendial method. So we clearly want to show equal to or better than the compendial method for sensitivity, which we are able to do. For very low titrations, it's the same type of protocol as other molecular systems in which there may be a need to do a 10-minute centrifugation step to reach the sensitivity of very, very low. But especially if you're trying to do a very fast test to see if you have a contamination of your raw materials, you're able to take that time a little bit versus more in process before you fill. And as far as the number of species tested, I know that all of the compendial organisms have been tested. And matrix interference with culture media, we have not seen any matrix interference from the culture media itself.
Thank you. Our next question is, when do you expect the new Microplasma test to be available? Dr. Laurie, would you like to answer this first?
Yeah, it's going to be released later this year, so we're pretty excited about it. We're actually starting to roll it out, I think, in the fall of 2019. Stay tuned.
And moving on, how long does it take to validate an alternative rapid sterility test? Dr. Miller, shall I answer this?
There's no hard and fast rule. Clients that I have worked with and I'm currently working with, if you have sufficient resources either in-house or at a third-party contract manufacturer or contract laboratory where the testing will be done, it's possible that you can run a full performance qualification within, I'd say, a 3-month period and leave a few more weeks to put all the documentation together. That assumes you have a robust validation protocol written beforehand, and that can take up to a month to do to make sure that everything is captured. So, I mean, the earliest, based on my experience, would be probably 3 or 4 months to have everything done. In some cases, it may take longer. You may have to go up to 6 months or more, again, depending on how many resources are available. This is a lot of work to do, and it's a commitment by companies to go this route. So as long as you have all your ducks in a row, this is not an insurmountable task, but you do need to have everything in place. And when I mention performance qualification, that would include all of your upfront primary validation work with standard organisms, your method suitability work with your product, and then whatever equivalent studies against the compendial method to be performed. So hopefully that gives you some indication of how long this type of work will take.
Thank you. Our next question is, what is the most common incubation time that the Baxter alert system is being used for testing cell and gene therapy products? Dr. Daane, would you like to answer this?
Yeah, that's a really good question. Typically, what I'm seeing, especially because of the age of the product needing to be released, generally within 7 days, many companies are incubating for 5 to 7 days. And they're focusing, again, on the clinically significant microorganisms.
And on the back of that, does bioMérieux provide validation support for implementing the Baxter alert system?
Oh, that's a good question, too. Yes, we actually provide a lot of the documentation to help customers with their validation. What's really good too is that we partner with an outside company as well, which is always really good to have, where they go on site and perform the IQ, OQ, and including temperature mapping. So we definitely partner with companies and help them through the entire process.
Thank you. Our next question. Can you use an automated method as an alternative sterility test? Dr. Miller?
Oh, the answer is yes, but let me make some definitions here. An automated method or an automated alternative method, even for sterility testing, is considered to be a method that is going to be comparable to the compendial test. It's actually, in many cases, considered to be an automated compendial assay. Okay, and I just need to clarify, though, that an automated compendial assay, if you deviate from the compendial requirements, which might include the required sample size in the compendial method, or even the incubation parameters, the time or the temperature from what is prescribed in the compendia, then you may need to do some additional validation type work. And here's an example. If you have a system that is considered an automated compendial assay, but you want to reduce the incubation time, then you would not necessarily have to go back and revalidate the entire method as you would with, say, a non-growth-based method. But I would expect a company to challenge the system to demonstrate that the detection level is going to be comparable to a compendial assay when a reduced incubation time is used. So when using a method that is considered or promoted as an automated compendial method, it is going to be your responsibility to understand if there are any differences in the way the compendial method is normally carried out, and if there are, make a risk assessment on any additional work that you may have to carry out. I also want to point you to one of the chapters in the European Pharmacopoeia that I had mentioned earlier in my presentation, Chapter 2.6.27 on cell-based preparations. That chapter does discuss automated growth-based methods, in which case, if you are following the compendial method, but you're using an automated system to run the compendial assay, that in that case, all you need to do is method suitability on the matrix to make sure you have no false positives or negatives, and then conduct growth promotion testing on the media that is normally being used in that automated method. So I would refer you back to that chapter as well, with the caveat that if you are making any changes to the compendial method, you may need to do some additional work with that particular system. Good question.
So thank you. I believe that's all the time we have for questions. I would like to take this opportunity to thank our presenters once again. That was Dr. Michael J. Miller, President of Microbiology Consultants, LLC, and Laurie Dane, Pharma Microbiology Scientific Director at bioMérieux. Thank you for sharing your knowledge with us. I would also like to remind our audience that you can view this and other webinars on demand by visiting biopharma-asia.com/on-demand webinars. And if you are watching this on demand, then please feel free to send your questions over to me at stephen.edwards@biopharma-asia.com, and we'll get those answers back to you. Thank you, everyone. Have a great day.